Hello everyone. I thought I'd do a quick overview on this research poster presentation in a line for the UKKW UKKW 2023, the UK Kidney Week seminar set to take place, and where this research presentation is linked to some works from originally from 2003 2004, and where this work is an overview in looking at observing chemokine and chemokine receptor expression in acute allograft rejection, a retrospective on human transplant immunobiology and laboratory practice. And so this research poster presentation will have an overview where there will be some where this works will look at an intro, some aims and objectives, looking at sort of the immunobiology and sort of the intricate aspects in around antigen presentation, and antigen presentation in context of transplant, transplant immuno transplant 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 sort of practices, and also in around allograft transplant rejection, looking at sort of transplant tolerance and in line with the chemokine receptors and ligands, and looking at chemokine receptors and leukocyte sort of trafficking in context of acute allograft rejection, or AAR. And there will be in this in this research poster presentation, there's an overview in around looking at chemokine receptors identified in human allograft rejection, and then there'll be some, and then there will be some, then there are some sort of conclusion or concluding aspects on this as well. So, to begin with the intro, renal transplantation is a preferred modality for recipients, or has been evidenced as a modality for recipients with end stage renal disease, or ESRD, and at the same time who sort of make good surgical candidates, but also where there are good surgical risks, where there are good surgical aspects, and where recipients are able to comply with chronic immunosuppression therapies, or IST. And clinical transplantation has generally advanced significantly, with most transplant institutions reporting 1 year allograft survival rates of better than 80%. Nevertheless, or however, a rejection and a progressive loss of allografts in this context, for those who are hearing the terminology initially, allografts is sort of a one sort of terminology used in a line to transplant, in line to where organ transplants have taken place. So the progressive loss of allografts over time have been evidenced as a continuing issue. However, at the same time, over the last decade, many reports of chemokine expression in context of clinical or experimental allograft rejection have been highlighted, and within this research presentation, there are several citations in linking the literature. However, there are at the same time often numerous ligands for a given chemokine receptor, and a specific chemokine receptor plays a significant role in the allograft response. Has become increasingly has been has been increasing become increasingly important. So the aims and objectives of this works was to seek out and to provide a retrospective overview observing chemokine receptor expression in acute allograft rejection, or AAR, in context of human renal transplantation and transplant immunobiology. So, in context of renal transplant immunobiology and antigen presentation, then organ transplantation has a retrospective history, or a long history, beginning with skin autografting in India during the 6th century BCE, and this procedure entered Western medicine during the early Renaissance, and is described in a text on the restoration of the nose, lips, and ears by works by Gaspar Tagliacozzi from 1545 to 1599, and in 1906 Matteo Jabulé carried out one of the first attempts at human renal transplantation, and Jabulé intriguingly used pig and goat kidney, anastomized or joining to blood vessels of the arms of patients or recipients with or those who are diagnosed with forms of chronic renal failure or kidney failure, which functioned for approximately 1 hour. And it was in 1911, Hammond and Sutton of Philadelphia performed the first human-to-human kidney transplantation or renal transplant with transient success. And it was in line to this and sort of looking at acute allograft rejection, or AAR. In AAR, acute cellular rejection is mediated by lymphocytes which have become active, alloactivated. So alloactivated means sort of automatically activated against donor transplantation or transplant antigens, and in vivo or in vivo stimulation of alloreactive T cells takes place primarily in the peripheral lymphoid tissues of recipients through intragraft sensitization, and which can often occur. And the strongest stimulus is provided by donor dendritic cells, also referred to as passenger leukocytes in the allograft, which will or can enter the circulation and end up on lymphoid tissues of the recipient. The dendritic cells at the same time function as antigen-presenting cells, and they provide a strong stimulus of human leukocyte antigen, or HLA class II reactive CD4 cells, which can stimulate the growth and differentiation of HLA class reactive CD8 positive cytolytic lymphocytes. Alloreactive lymphocytes also enter the circulation, and they react, and they will or they can react with allograft vascular endothelium, and the primary target of the initial stage of cellular rejection. So in line to so now sort of in line to looking at transplantation tolerance, but also in linking with chemokine, in linking with chemokine receptors and ligands. So of approximately 18 chemokine receptors, at least 3 retrospectively had appeared to have played an important role in the rejection in experimental models. CCR1, CXCR3, and CCR5. And in experimental research models, the chemokine receptor antagonists are more effective when used with low dose calcineurin inhibitors. So calcineurin inhibitors are sort of first or second line generation of immunosuppression regimens that are prescribed to those who are prescribed to those who have solid organ transplants, and CNIs such as, for example, cyclosporin A, or CSA, and other CNIs, so again, in experimental research and models, the chemokine receptor antagonists are more effective when used with low dose CNIs, than when administered as monotherapy. So monotherapy being immunosuppression alone, where in context of transplant practices, CNIs alongside with either a second form of IST or immunosuppression, or part of triple therapy, being part of including for example steroidal medicine, therapy or pharmaceutics as well. So developmental programs are in place or have been in place for antagonists of CXCR3 and CCR1, and it had been transparent from retrospective investigations that blockade of chemokine receptors have become essential topics of an addition to therapeutic prevention of acute allograft rejection, or AAR. The combination of clinical expression of certain chemokine receptors during the rejection, with the compelling evidence from rodent studies using knockout mice, and or neutralizing monoclonal antibodies, has led to the consideration of therapeutic targeting of CXCR3, and CCR5. To this end, pharmaceutics have been developing small molecule antagonists and humanized monoclonal antibodies that have been tested in preclinical models, either alone or in conjunction with IST or immunosuppression. The first peer-reviewed data on sort of in around chemokine receptors and chemokine receptor ligands, in non-human primate allograft recipients have been available following research since 2000, since 2003. In line to this as well, this research poster presentation has a sort of a schematic which sort of outlines the chemokine receptors and chemokine receptor ligands. So for each chemokine receptor, you have you have a ligand that allows a binding a binding of chemokine receptor to ligand. And so several important points have emerged from investigations based on mechanistic based on the mechanistics. And first, targeting of a single sort of chemokine is typically ineffective in prolonging allograft survival. An example of this is a general principle is that the lack of efficacy in targeting of macrophage inflammatory protein, and whereas targeting of receptors such as CCR1 and CCR5 is of therapeutic significance. Second, chemokine receptors differ in the importance as targets in allo responses. Chemokine receptors such as CCR1, CCR2 primarily promote the macrophage recruitment to an allograft, such that the targeting in knockout mice has only a modest effect on allograft survival. And third, the effects of containment of immunosuppression therapy, or particularly in specific immunosuppression, can modulate some of the rather black and white outcomes of chemokine receptor targeting in otherwise untreated mice. In the case of chemokine receptors, with the most impressive experimental data to its role in allograft rejection, expression of CXCR3 and its ligands is closely associated with acute allograft rejection, or AAR, clinically. The various stimulatory and inhibitory pathways involved in these activation cascades are providing are and have been providing insight into the design of potential pharmacological agents. So in line with this, a bit more about chemokine receptors and leukocyte trafficking, because it's the leukocyte trafficking the leukocyte trafficking in context of acute allograft rejection, where chemokine receptors have been evidenced as being most important. So in renal transplantation, the occurrence of one or more episodes of AAR, acute allograft rejection, is a major determinant of graft survival. Most episodes of acute allograft rejection are caused by cell-mediated processes and require the infiltration of allo-activated T cells into the engrafted organ. Over 14 inducible chemokines and 20 inducible chemokine receptors have been identified to date, and in vitro studies on responses of activated T cells to inflammatory chemokines suggest a substantial redundancy in this system, which with multiple chemokines produced at tissue sites binding to multiple receptors on circulating leukocytes. Chemokines binding to receptors on leukocytes mediate the behind the scene plays between antigen presenting cells and host T cells in lymphoid tissues. The actual here and now to propagate an immune response that actually destroys an allograft, and seems apparent sort of smoldering inflammatory responses seen clinically and referred to as chronic rejection. So in this research poster presentation as well, there is an overview of chemokine and chemokine receptors identified in human acute allograft rejection, wherein on one side there's a sort of a list a sort of an overview or a list of chemokines, and then where in literature there are some findings against those of those particular chemokines, and then is the citation. There is a citation around those. So, for example, going down the list of looking at chemokine receptor CCR5, CCR5. For example, the findings around CCR5, at least retrospectively, have been that CCR5 expression is associated with mononuclear cell infiltrate in acute allograft rejection, and there was a citation by works from 1998 by Etner and team, and in around the in around this this yeah this sort of overview. There's the list in this research poster presentation is based on chemokine and also chemokine receptors, and so in sort of conclusion, or in concluding, in a clinical sort of representation by Hancock and contemporaries from 2002, 2003, and sort of more earlier T cells within, for example, bronchiolar lavage or BAL of lung transplanted recipients undergoing rejection episodes. Evidence of CXCR3 positive was exhibited with a strong in vitro chemotactic response to IP10 and graft infiltrating macrophages expressed IP10, and alveolar macrophages also expressed and secreted IP10 at levels capable of inducing chemotaxis of a CXCR3 T cell line. IP10 production was upregulated by interferon, and high levels of CXCR3 ligands were verified within BAL fluid or fluids of recipients with rejection episodes. And overall research retrospectively informs that there are direct effects of chemokines on recruitment, proliferation, and activation of vascular smooth muscle and other non-hemopoietic cells, in addition to mediation of leukocyte recruitment. Induction of CXCR3 and IP10 mRNA has been associated with acute allograft rejection, aligned to human renal transplantation and transplant immunobiology. The research poster presentation has a few acknowledgements and sort of a few dedications as well. So that draws this overview to an end. This research poster presentation overview is and should be available on the RPSG, the Renal Patient Support Group RPSG ResearchGate page, and also on other RPSG social other sort of social RPSG and sort of KDOS social media. Thank you for tuning in.